Good morning to those on the West Coast, good afternoon to those on the East Coast, and good evening to those of you across the pond on the other side of the world. Thanks for joining us today. I'm Laura Esserman, Professor of Surgery and Radiology at UCSF. I lead the I-SPY family of trials, and I have the privilege of being on the CERSI board. I'm joined by Dr. Peter Marks, who's the Director of the Center for Biologics Evaluation and Research at the FDA, and who will give us a keynote in just after this panel. We are hoping to have a very lively session and are really fortunate to have a terrific panel. First up is Dr. Gideon Blumenthal, who's the Vice President of Global Regulatory Affairs for Oncology at Merck, but until last January was the Deputy Director for Oncology at the Center of Excellence at the FDA, so he knows both sides very well. Dr. Martha Donoghue, who's the Acting Deputy Director of the Division of Oncology 2 in the Office of Oncologic Diseases at the FDA, and the Oncology Center of Excellence. Dr. John Tsai, who is the head of global drug development and chief medical officer at Novartis. And last, but certainly not least, we have Dr. George Yancopoulos, who's the co-founder, president, and chief scientific officer at Regeneron. So the ground rules for this panel are the following. I'm going to lead a round of questions for the panel to start us off. And Peter Marks is going to follow by leading the Q&A session with the audience and our panel. So get your questions ready and submit them as we go along. Just put them into Slido and we'll get to them. So for our panelists, I'm going to ask each of you to limit your answers to 2 to 3 minutes. A timer will go off after 3 minutes so we can wrap up. And then after the Q&A with the audience, we're going to do a quick lightning round. Okay, so I'm going to start off, and first I'm going to start off with Gideon as going down the panel. We're going to go in that same order. And Gideon, fitting that we start with you first, we'd like to know what you think is the biggest misunderstanding that pharma and FDA has of each other, and what are the barriers that we need to break down to make oncology drug development more effective?
Yeah, thanks, Laura, and great to be here. I would say, I spent over a decade at the FDA, and now I've been in industry for almost a year. So I think I have a better understanding of both sides after this eventful year. I would say on the industry side, the biggest misconception of the FDA is at times there can be a perception that the FDA are essentially overly bureaucratized. They don't have the sophisticated knowledge or experience to make complicated drug development decisions, and that they don't have the requisite expertise in-house. On the FDA side, sometimes there's the impression that, which is somewhat true, but industry is beholden to profit and shareholders, but ultimately most people go into drug development and industry to really move the needle for patients and get better therapies out on the market for patients. So I think in that sense, incentives are aligned between FDA and the industry, although obviously differing missions ultimately. As far as the barriers that we need to break down to make drug development more effective, I really think that, at FDA and oncology, I mean, it's really, I think, the gold standard in terms of drug regulation. Globally, it's led with so many initiatives and like real-time oncology review, the Oncology Center of Excellence, Project Orbis, breakthrough therapy, accelerated approval. And we just have to, continue letting the oncology office do its thing and lead the way, not let it, if other therapeutic areas, for example, have other complications or challenges, we have to let oncology really continue to strive and sustain the excellence that's transpired. I think sort of codifying some of these initiatives that have been propagated in the last decade, like Orbis and real-time oncology review, to take it sort of to the next level would be very, very important for the industry, both in the U.S. and globally. Thank you.
Okay, great. All right, Martha.
Thank you so much for having me here today. And I think I'll probably touch upon some of the items that Gideon already mentioned. But in terms of misunderstandings from pharma, and I think, I see this a bit more from companies that are outside of the US than in the US, but we often see development programs that closely resemble or are almost identical to development programs for indications or classes of drugs that have already led to previous regulatory approval. And with that sort of is the implicit assumption that the pathway to approval is static and that we're not amenable to different types of clinical development programs. And this isn't the case, and we certainly encourage interaction and discussion with sponsors to ensure that the drug development pathway moving forward is the most efficient and thoughtful way program that it can be. And that doesn't mean that we will always agree with proposals from sponsors, certainly, but we're very interested in having robust dialogue how we can change things in the future to make drug development programs go faster so that we can deliver safe and effective drugs more quickly to our patients. When providing advice to stakeholders, we take into account a variety of factors, scientific advances, prior clinical trial data, treatment landscape as it stands at the time, and patient needs. Just a few examples of some of the evolving approaches that we've kind of led the way for, I think, in oncology is the tissue agnostic drug development, for example. Also, we've been adopting and using over the past few years this model-informed drug development program to facilitate development. And this program uses exposure-based biological and statistical models derived from preclinical and clinical data to help improve clinical trial efficiency, optimize dosing, and things of that nature without the need for a dedicated trial. We've also been on the forefront of looking really closely at eligibility criteria, try to make sure that they are as broad as possible when warranted in order to make clinical trials accessible to patients. Regarding the barriers that need to be addressed, I think we've had a lot of discussion already in this conference about the need for increased collaboration, continued work to break down communication barriers so that all stakeholders, including FDA, are communicating effectively with one another. That's absolutely vital. We need to increase global collaboration, as Gideon has said, and we've had some wonderful experiences with Project Orbis, with collaborative reviews, with international regulatory agencies on applications we're looking at. And I think that's been very fruitful, has resulted in some, I think, faster approval processes in other parts of the world. And I think we need to keep doing things like that and refining and thinking outside the box a lot more. And I think finally, we need better, more equitable access to data from completed clinical trials amongst all stakeholders and collaboration for developing new biomarkers for more efficient drug development.
Perfect. 3 minutes exactly. So I think you would say more communication more often, and people have access more than they think they do. All right, John, from your perspective, what do you think?
Yeah, thanks, Laura. Thanks for having me on the panel here, and good evening from coming over from across the pond here. So maybe to start, what I would say is I actually don't think that there are big misunderstandings, and that's the way the question was worded, was what are the big misunderstandings between FDA and pharmaceutical companies. But what I do think is that there is new science and technology that's moving at lightning speed, and our drug development and approval paradigms are built based on the history of what we have advanced. And so it's something that was developed a while ago. So it's moving at a much slower speed than currently what science is advancing at such a quick speed. And as we see science and technology emerge, we recognize the importance of working together, as I know was stated earlier. And how can we actually move in partnership to move together in a timely manner? And the current system isn't just set up for almost what we call pseudo real-time interactions. For example, as we have these adaptive trials where we have seamless designs, we're getting information and results on a timely basis where we're turning around and changing how we recruit patients, how we actually are adapting our trials, and the exchange with the agency isn't built for that. So what I would say is that we have a common goal, which is to protect the same as the FDA, which is to protect the public health and to uphold the evidentiary safety and quality standards for new medicines, which is the same for FDA as well as for pharmaceutical companies. And how can we actually make that happen? I think that's the importance of what we need to do. And one of the things that we need to consider is, as we're getting results, that we're conducting these trials in different patient populations across the globe. So there may be different treatment journeys that we need to take into consideration. Maybe to touch up on one additional point here, which is, and we talked about oncology, but specifically in cell and gene therapy, the science is moving faster than the current regulatory paradigm. So on one hand, what we're seeing is big innovative steps that preserve T-cell stemness with the dual CAR T therapies. And at the same time, the same CAR transgene, we're seeing innovations in how we collect, how we manufacture, how we deliver these cell therapies with greater reliability and reduced turnaround. And as we're seeing these quick, rapid cycles, we need to be able to translate these innovations into innovative therapies where there's a regulatory framework to support the sustainable development of these paradigms without unnecessary redundancies. Maybe one last point I'd like to touch on is potentially manufacturing, where we're learning together. We're finding that sometimes the important changes happen after the approval, such as when we're looking at efficiencies or site expansions. What this means is that we really need to stay lockstep and not just till the time of approval, but well beyond that. So just to close, no big misunderstandings, but I think additional collaboration is needed for us to advance as quickly as science is advancing. I'll stop there.
Thanks, John. Okay, George, your turn.
All right. Well, first of all, I'd also like to give my thanks for being invited to participate here. And obviously a lot of the points that I was going to make have already been touched upon by everybody. But I do want to echo particularly what John was just saying, is that I also don't think that there's really major misunderstandings. But the way that I look at it is that there's very different roles and perspectives for the FDA and for companies, especially some companies. Martha told us some companies are too boilerplate in the way they're trying to do their drug development. But I think there's a lot of other companies, companies like Regeneron, that are constantly pushing the envelope and trying to have new innovative ways of speeding up the drug development process. And the job and the role of the FDA is to, I think, balance that and make sure that there's not, and to push back and to make sure that there's not too much rushing and that we're not doing things inappropriately that don't lead to, appropriate testing and validation of the various agents that we're doing. So I think it's about a balance and the different roles between the two. And I think that it is the role of companies in many cases to try to be pushing that envelope. And I think it's the role of the FDA to make sure that we're not pushing too hard and too fast and making mistakes along the way, because they have a broader perspective and experience and background than almost all of us put together. And I actually think that the system works. It doesn't mean that we don't have constant, lots of differences, lots of arguments, nor that we can't evolve and do things better. But that's what it's all about. And I think that that's what you need. You need people on both sides. I mean, that's why we have the companies pushing, and that's why we have the FDA making sure and trying to keep everything in line. And so that's what I think it's really about. And I think, continue to improve and maximize. That's all part of the evolution of the process. I also want to touch upon, some things that I've certainly noticed now during the whole COVID-19 pandemic. As many of you know, we've been trying to advance a whole assortment of different therapies for COVID-19. And the interactions, John touched upon real-time interactions. The extraordinary speed in which the FDA has been responsive for COVID-19 treatments during this pandemic is something that I think is unprecedented. I mean, the turnaround time where they can make decisions, where they can give their input, they can make decisions about us going forward or challenging some of the approaches that we're making. I only hope that we can keep it up and extend that beyond and outside of just the COVID-19 treatments. I don't know if the resources and the capabilities are actually there to do it, because obviously everybody's been mobilized to try to deal with this horrific pandemic. But it is extraordinary to see what can be done and the speed by which things can move when maximum interactions occur and are allowed. I don't think it's the FDA's fault if they won't be able to give that time, that extraordinary type of speed in terms of responsiveness and so forth, when you move back to a state where they're dealing with, all therapeutic areas and so forth. But it's been extraordinary to see when they do put their resources and focus them on this COVID-19 pandemic, how fast things can actually happen. And I don't know what it would take to extend that capability and those resources more broadly.
All right, so let's delve into that. And certainly I think that both COVID is an example where industry came together, and I think in some cases the academia and the other trial designers have come together to try and speed things up. But we need to take these lessons into oncology and cardiology and in HIV. Early endpoints have certainly accelerated the development of many, many new agents that reduce mortality and morbidity and have really accelerated cycle times. But so in oncology, how can, as we develop new drugs and successful classes of agents emerge and then proliferate, how can we work to develop drugs to rapidly test which combinations can increase response and decrease toxicity? You can use any example you want. The PD-1 are a perfect example. Do we need 20 of them? Why is this necessary? And what should we do about it? Gideon, we'll start with you, since you have are one of the leader positions. You might have a different view than, say, George might have. Go ahead, Gideon.
Yeah, I mean, it's a great question, Laura. And I think you've kind of led the way with the I-SPY 2 program in sort of spearheading the adaptive design concept, validating pathologic complete response for high-risk breast cancer as a, potential reasonably likely surrogate endpoint. All of the great things you have done, with I-SPY 2, I think those lessons could be learned more generally beyond breast cancer. I know you've used this during the COVID pandemic crisis, but I think specifically in cancer, I think combining pathologic endpoints as well as changes in circulating tumor DNA. I think this technology is really here. It's real, and we have to figure out how to incorporate these technologies into drug development. For the longest time in oncology, we've been using RECIST 1.1. And everyone acknowledges that the limitations of RECIST, but we're sort of stuck with it as a historic artifact. But can we use novel imaging modalities, new biomarkers to monitor the patient, to get a better handle on what truly the disease is doing, like has been done in HIV. That being said, and I think you were alluding to this, I do think that we should maintain high standards. So, how many PD-1s do we need? That's sort of a philosophical question. I can't answer that. But for the innovative companies that have developed the evidence, I do think it wouldn't make sense to necessarily lower evidentiary standards to flood the market, to sort of commoditize these agents.
Sorry, Martha. I think that, sorry, I had put myself on mute. Thank you, George.
Thanks, Laura. I do that a lot. So I think Gideon touched upon sort of the use of earlier endpoints that we've long used in oncology to promote, faster drug development based upon imaging endpoints, essentially, such as durable overall rate and progression-free survival. And I think there is a great need to develop and validate new biomarkers to make drug development combinations faster than it is now. And even for those early endpoints or biomarkers, if they haven't been fully validated, I think they can still be used in early randomized phase 2 trials, for instance, to help inform go-no-go decisions. And I think we don't see enough of that. At least speaking personally, I see a lot of single-arm development early on, and an immediate transition to a large phase 3 trial. And I think having earlier, maybe platform-based randomized phase 2 trials looking at earlier endpoints would really go a long way in helping not only to inform, selection of new combinations and new novel drugs to be kind of moved forward in development, but also could also help facilitate the validation of early markers such as circulating tumor DNA so that they can be validated and eventually used as endpoints to help support regular approval or accelerated approval. We're working really hard in efforts to try to promote the use and validation of new biomarkers to support drug development. I just wanted to mention briefly the CT Monitor project, which is the public-private partnership led by Friends of Cancer Research, which is looking at circulating tumor DNA across studies to see if changes in circulating tumor DNA reflect responses to treatment. It is, the project aims to validate these blood biomarkers to see if they can be used and how they can be used in drug development. Stage 1 of the project is complete, and this looked at associations between circulating tumor DNA and endpoints such as response rate and progression-free survival and overall survival in non-small cell lung cancer. I think we learned a lot, and there did appear to be some associations between large changes in circulating tumor DNA and other endpoints that we're more used to using. We also learned a lot about sort of the caveats and combining data and analyzing it that I think we can take and move forward into stage 2 of this project. I just wanted to mention briefly when we're thinking about combinations and development of combinations from a regulatory standpoint, it's very important to us that the drug development program characterize the effect of each drug used in the combination so that, for example, for a 2-drug combination, we need to have the confidence that both drugs are needed in order to achieve that observed treatment effect. And there's no one-size-fits-all approach to doing this, so we do encourage people to come talk to us about their strategy for showing that if their drug development program involves two drugs in combination. And I'll just end there for now.
And I would say, Martha, from the FDA standpoint, once you've got a good link between your early outcomes and long-term in a drug by class, that might open the door to saying, okay, well, the next one coming in, you got to show the early endpoint, and then maybe putting some emphasis in larger phase 4 and things like that to establish things like safety, or there could be other ways to speed things up, perhaps.
Absolutely.
Okay.
Yeah, absolutely.
All right, John, what's your opinion?
Yeah, thanks, Laura. I had many similar points, but maybe I'll take an industry perspective on how we're thinking about designing clinical studies and the ways that we think about accelerating potential development programs in oncology. One of the things that we've done is we've set early alignment with joint seamless teams integrated to ensure that we have the both early and late development teams. So once we have the information, the data that's available to go transition quickly into full from phase 1 directly often into phase 3, which you actually don't often see happen in cardiovascular programs, given that the benefit risks that we're seeing. So that's one of the things that we do is set very early alignment between the teams. Secondly, I think is around ensuring that we take calculated risks on leveraging endpoints and data, not only from our own programs, but also from the competitive programs that are out there, because what you'll see is the oncology landscape is so competitive that often very similar mechanisms of action, and you can actually use the insights from competitor programs to actually help design those trials. And probably third, in terms of accelerating our designs, is what was mentioned earlier, is ensuring that we document the evidence in terms of the contribution of components. And I think Martha was alluding to this just earlier, is I think this is one of the critical innovative areas that we need to think about. Is often in oncology, we're looking at combinations and the importance of ensuring that we not only know the contribution of one component, but also the additional components in the studies. I guess one additional consideration would be adaptive designs, and I know we'll probably have some discussions around adaptive designs, but I think this is another area where we could accelerate approvals and exploration of oncology drugs. And one example of this is the PanCan study, where we're looking at pancreatic cancer, which has been such a difficult area to explore. But when you can actually look at adaptive designs across potentially multiple agents as an umbrella study, this helps not only decrease the number of patients, but also exposure of patients to different treatments. And my last thought on this topic would be really around biomarkers and precision medicine. I think Martha actually outlined this really well, so I won't explore or go into this in further depth unless there are questions. So maybe I'll stop here, Laura.
Okay, thanks. And George, certainly you have a very innovative approach in your own development pipeline. So what are your thoughts as you sort of think about what's in your pipeline?
Yeah, once again, a lot of these points have been already touched upon, but it does speak back to this balance between pushing the envelope, which is what many of us in the industry are trying to do, and balanced by the FDA, making sure that when we're trying to have an innovative molecular readout or some other proposed surrogate or biomarker, that it really is a useful and a validated one. So I think it is important to have the balance. And from the FDA's perspective, especially for full approvals in many settings and in the frontline settings, of course, they're maintaining overall survival as always being the final standard, which obviously limits the number of large, full programs. And I do think it is worth pointing out that it may not be appropriate to always have the same standard for all the different classes of drugs. And you mentioned we could use examples. I am going to use PD-1 as example. We've heard RECIST has already been maligned and talk about other kinds of endpoints and molecular markers. But I think that for the PD-1 class, actually measuring tumor size and responses therein may be the most powerful and nearest term way to really evaluate and compare these sort of agents. I mean, we've certainly shown, we believe that continuous tumor burdens and responses, when measured that way, and not using traditional RECIST approach, but by measuring actual continuous tumor measures, may be the most powerful way to compare agents like in the PD-1 class when responses are durable. Why are responses not thought to be the be it and the end all? Because of the question of durability of response. But when you have a class of a drug like a PD-1 that is shown repeatedly that the response is so durable, I think you can consider moving to using things that are assessing the actual impact on the tumor, like continuous tumor burdens, not only for early stage analyses, but actually for full approvals. And the question is why? You brought it up. How many PD-1s do we need? Do we need many PD-1s? I would actually argue that we do need several PD-1s. Okay, and why is that? I think it's because so many of us believe that the future in so many cancers is going to be combinations. I mean, this is what you're doing with the whole pioneering I-SPY program and so forth. We do have to figure out ways to more efficiently test combinations. But if you think about the cost to society, if there's only a limited number of PD-1s, and if you have a whole slew of companies developing potential combinations, if you have to pair one company's next agent with another company's PD-1, the cost to society becomes incomprehensible and unsustainable. Whereas if we allow multiple companies to have this foundational PD-1 therapy that so many of us feel will be foundational to so many of these future combinations, then one can imagine an individual company that controls the price of both agents, allowing these to be made much more affordable to society than if you have to rely on two different companies. So the bottom line that I'm saying is that I think we have to consider different endpoints for different classes. I think controversial endpoints like responses can actually end up being full approval endpoints for a class if that becomes validated over time. I think there's huge value in many cases when you have a foundational therapy like a PD-1 to have many different PD-1s because they're going to become the basis for approvals. And of course, we're going to need new approaches to test all these combinations and try to really understand which combinations really are going to have the biggest contribution to patients going forward.
I agree. And to move them forward earlier in the disease course, where we know they're likely to be much more effective, and not be so afraid to try it in earlier stage disease where there are more robust early endpoints like complete response.
Yeah, I mean, absolutely. I mean, obviously, and you've been pioneering this area, and many of us are following your lead here. I think that instead of looking at the late stage, where the impact of any individual drug is probably less and harder to see, it's probably more powerful to go earlier, where the impact of almost any drug we know is greater. It'll be more measurable. It'll be an easier way to get a first pass result that shows that that combination is viable. But we all believe that it's all going to be the future in combinations on top of certain foundational therapies. And right now in immunotherapy, PD-1 really seems to be a foundational one, and one that people need to figure out how to have access to.
Okay, great. All right. So before we go to the audience questions, how about a 1-minute answer from people? A lot of talk about real world evidence and patient reported outcomes is playing a meaningful role in the assessment. Now, especially as we start putting combinations together and trying to figure out how we can have non-chemo combinations, where do you believe this is going to have a role or should have a role going forward, and how can we make this more sustainable going forward? Gideon, about a 1 minute-ish.
Yeah, so, I think that RWE and patient-reported outcomes, I still think in oncology, we're a ways away to using these tools for confirmatory evidence. Obviously, they have been used, well publicized as supportive evidence in various, BLA, NDA submissions, but we still,
You're not a believer, not yet.
Unfortunately, not yet. Yeah.
Data, you want to see data. Martha.
I agree with everything Gideon said. We do have, examples of use of clinical outcome assessments to support approvals. Most recently, selumetinib for the treatment of pediatric patients with neurofibromatosis and plexiform neurofibromas. We looked at reduction in tumor volume there, but I also looked at improvement in tumor-related symptoms, and that really did help, I think, get them over the edge to approval there. For real-world evidence, we have a couple approvals that have used supportive data. The real-world evidence, I think you would know better than I, the palbociclib example, which was the approval was extended to men, based upon clinical trial data, in addition to some Flatiron data and IQVIA data. So I think the potential's there. I think we're in the infancy as far as real-world evidence is concerned. As part of the Oncology Center for Excellence, we've now established a project and program specific for looking at real-world evidence proposals, which I think will help us greatly in assessing these proposals, being more transparent, and working together with the community to try to leverage the potential real-world evidence. But we're not there yet.
And that's one of the reasons why we're trying to get these one source tools. If you can, in your course of care, collect the same high quality data, and you're generating evidence as part of the standard of care, or your byproduct of care, that would change everything. So that's really, I think, an important place to go. John, are you incorporating patient-reported outcomes in all your trials?
I wouldn't say we are incorporating RWE in all of our trials, Laura, but I do think that there are opportunities, and I think Martha's outlined, there's only been a handful of approvals using real-world evidence. But I do think that there's opportunities in rare diseases and rare tumor types where it's difficult to collect that data. We did use real-world evidence in the approval for Tabrecta, which is our MET inhibitor. And what we saw was that there was only 3 to 5% of non-small cell lung cancer patients who have this MET mutation. So using this real world evidence, we were able to use this as background information across multi-countries to collect baseline and demographics data on these patients to help the approval. So I would not say that was the evidence for the approval for Tabrecta, but it does give you some comparison, particularly when it's difficult to collect data in the rarer tumor types. So agree with what everyone else has said, but I would say I would leave a space there for some of these more difficult to collect data on some of these rare tumor types.
So George, a lot of your pipeline involves things that hopefully will allow us to drop some of the standard chemotherapy. Do you think patient-reported toxicity is going to be the key to helping you move some of these combinations forward?
Well, I'm not sure. Well, let me echo some of the other people's thoughts on real-world data. Other than rare examples, I really do think that we have a very, very, very long way to go. But it speaks to a much bigger issue of how much healthcare information we are throwing away as a society.
That's right. Yep.
And what a waste it really is, especially with how much we spend on healthcare, that a little bit more investment could contribute and then save so much in terms of healthcare costs. And we've already touched upon it. I think the only way real world data will end up making a difference if we do what you already touched upon, if we incorporate into standard of care the getting and the entering of regular high quality data. I mean, this depends on, right now it is imaging. Maybe if we develop new surrogates and all that, we can incorporate those. But right now, the only way we could really use powerfully the real world data, if we incorporate much more powerfully widespread use of imaging, regular imaging, incorporation of the imaging standards, and into record incorporation. I mean, this just speaks as a nation, it's almost criminal, okay, that we are not doing this, how much it's costing us, and how it could be helping the healthcare burden. And let me just give one example of why. I know there's rare examples, the Flatiron approaches or whatever, but we all know, you can't depend on these sorts of things. I mean, for all of us, and I'm sure the FDA has more experience than anybody else, when you depend just on investigator-read scanning images, you get different answers than if they're read blinded. Okay, that just speaks to me to the absurdity about thinking about going to the doctor record and the patient record and in a non-rigorous manner, trying to use that information in a very, very useful way. So I think we have to go in the direction that you're talking about. We have to change the standard of care. We have to invest a little bit more. And in the long term, it'll save us millions and billions and trillions. And it's the old story about an ounce of prevention is worth a pound of cure. Yeah. But as a society, we repeatedly have not shown that willingness to up our investment now to save ourselves enormous headaches in the future.
Absolutely could not agree more. The lesion in the system is our clinical care systems. We should have one system for care, which will work for research as well.
And information management, absolutely.
That's absolutely right. Okay, I'm going to turn this over to Peter. Peter, you are on.
Oh, thanks very much. Thanks. That was a great discussion from everyone. So we have about a half hour for questions, and some questions have come into from the audience that are all over the map here, which is great, but they're relevant to this discussion. Maybe we'll start with one that actually is, there are some that are very regulatory, some that are broader. Here's one that's interesting that goes back to the PD-1 inhibitors. Some people are interested in how we might get to a point where we could get to a public domain PD-1 inhibitor so that anyone who is developing combo therapies could have one available. Any ideas there or thoughts there about how to deal with that issue? It's one of comparators, I guess.
Who wants to take that one on?
And thoughts from anyone here? Any ideas?
Yeah.
I mean,
or go ahead.
I'll take it on. I think that if we could sort of carry it out at some point, it makes a lot of sense. Unfortunately, as we were just talking about, in our current healthcare system, and while we do these things that make sense don't always get implemented and utilized. And that's why I think that in the short term, it's very important for several individual companies that are trying to move their combinations along to have their own validated and approved PD-1. But I think, if there was a gold standard that would be available to everybody, that everybody could control the price and all this, but I don't know how we make that work in a society, because the other thing that we really have to understand about our society is the way things get done is when people are highly incentivized to get them done. Okay. And so we cannot destroy the system that creates the great incentives that lead to the major efforts that people are willing to literally bet hundreds of millions, if not billions of dollars on. So these are very, very complicated issues. And I think that, we really need to think about how to solve them.
Martha, you want to comment on that? Or whether you think that's even remotely feasible?
the question is definitely not within my pay grade, but I think it's a fascinating question to answer. I do think it's something that, we as a society would have to decide is our priority and put the funds and infrastructure there to kind of back it up. Maybe through use of, through NCI, maybe some sort of acquisition of enough PD-1 to be used by investigators. It would probably be on a competitive basis though, and maybe not universally available. But I think it's a great question. I wish I had a great answer.
Hey, Laura.
John.
Yeah, it would be great if I could just
John,
add a couple. Thanks. Can you guys can hear me? Okay. Yeah. Just give me a nod. Yeah. So.
Yes,
first, I guess one of the thoughts would be the underlying assumption that the PD-1s are very similar in efficacy and similar as a class of agents. And I guess that's the underlying assumption for this question. Noting that PD-1s are similar, PD-1s, PD-L1s across this schema, then maybe there could be a way where we could build a consortia and there would be agreements, given that it is a backbone therapy across industry. Potentially that is one approach, but I think without having everyone working closely together with this type of approach, I think, one of the major expenditures that we have is buying competitor drugs. And is there a way for us to actually find ways where we don't have a more collaborative approach in terms of investigating new agents? And I think you would have to get everyone within the industry to sign up for that. But that's maybe something that's forward-looking as a potential for exploring new ways of drug development.
Well, I just have to add to that because I think it's a great point that if that was the assumption, which I sort of misunderstood, I assumed that we would try to get, the best PD-1. I don't think the evidence actually suggests that these PD-1s and PD-L1s are not created actually equally. Just think about it. The first PD-1 of Devo has, after like 10 years, not been able to demonstrate convincing monotherapy activity in first-line lung cancer. What does that mean? Does that mean it's just like every other PD-1? And same with several of the PD-L1s. I mean, it looks like there's only three in the entire class that have been able to demonstrate convincing monotherapy and first-line activity, including one of ours, I should mention. But I think that is a huge issue, is that if you're going to pick a PD-1, you better damn well pick the best one. And this goes back to this whole notion that I said about before, is that whenever we try to get government to do certain things, I mean, I think government is a great watchdog. Government is a great regulator. But when we try to create government to drive things and to take away incentives from industry, then you get less done and things get done less well. So these are all things that we have to try to anticipate and think about. I think it's like everybody said, like Mart said, it's a fascinating question. In some ways, if you could do it right, it might be a great contribution. But it seems like it'll be very complicated to figure out how to do it right. It's not that simple.
Great. Thanks a lot. Those are great answers here. There's a question that a couple people have asked, which goes to comparative effectiveness, actually almost asking for comparative effectiveness up front. One of the questions, there are a couple of different ones, which all boil down to, should FDA require head-to-head trials for new therapies for a given entity, rather than just allow non-inferiority trials to get things on the market? Interesting, or how do you position requirements for head-to-head studies? So maybe we'll just take a round of people. Maybe I think people sometimes have allergic reactions to comparative effectiveness studies, but it looks like George wants to go right ahead. Go ahead.
Yeah, we've actually done comparative effectiveness trials, which have actually worked in our favor. So in many settings, so we're actually in theoretical ways very much in favor of that concept. But the same people who are asking to get comparative data up front are the same people who don't want to pay for drugs. Okay, and what they have to realize that these comparative head-to-head studies and these upfront studies, that if they're really powered appropriately to actually show that things really are within a small range, similar or not similar, are the largest of all studies. Okay, which cost the most. So if we want to do that, then the cost of drugs are by nature going to have to go up and up and up. So this is what people don't understand. You can't have everything. You can't have it cheap, and you can't have everything directly compared to each other. You can't do everything. And it's all a matter of solving this complicated equation of incentivizing individuals and companies and so forth to do the right things to get the best sort of answers at a cost that society can actually handle. So it's a great notion. Okay, it's that nobody will want to pay for that, and nobody will want to pay for the drugs at the end of the day that will take trials that instead of taking hundreds of millions will cost billions and billions to undertake. So people have to understand that.
Yeah, I'll take moderator's privilege to say that when I hear people talk about this, the way they argue for paying for them, George, is eventually that they do agree with you that it's going to cost more initially, but they then say it will cost the overall system less in the long run because you'll be eliminating a lot of ineffective therapies that are actually shortening people's lives or harming care. So I think that would be potentially, if I'm not advocating the position, I'm just as a person who's heard this argument, that's what I think they would argue, is that ultimately doing such may help reduce healthcare costs or lead to much better outcomes. But any other panelists want to comment on this? Looks like both John and Martha. Go ahead, Martha.
Hi, thanks. I just wanted to comment real quickly and point out that, in terms of approval standards, we don't have a comparative efficacy requirement to approve drugs right now, just so everybody's on the same playing field here. We just have to show that drugs are safe and effective. And we've done that in oncology in many ways, a lot of times now with the accelerated approval pathway, looking at durable response rate, of course, in a refractory population for approval. But we often, do approve based upon randomized trials, looking at the drug or combination versus what we consider to be standard of care at the time. In order to receive approval that way, of course, the investigational drug has to be shown to be an improvement over that control arm. The non-inferiority method of approval is actually very infrequently used because in order to prove non-inferiority, you need a very, very large clinical trial in general. And so we have a few examples of use of a non-inferiority design, but not that many. Lenvatinib is one example for HCC. There was a non-inferiority looking at that against sorafenib in hepatocellular carcinoma. And I think at least, my viewpoint is that, once we've decided a drug is safe and effective, the community and, does a very good job of determining over time how best, which drug might be the best drug for their patient, and kind of leaving it in the hands of the patient and healthcare provider community to some degree. And then, of course, in the post-marketing setting, additional clinical trials shed light on how best drugs can be used. I'm not saying there isn't room for improvement, though. There is room for improvement.
Great, John?
Yeah, I would agree with both George and Martha in their comments. But one additional consideration, in addition to economics, would be, would we actually stymie innovation? And the reason I mention that is, as we think about developing clinical studies, if we're developing studies for superiority, it is a high bar. It's a larger study, and it costs a lot more to conduct the study. And if that's the case, then it will require that we put much more effort into how much we're willing to take on that risk in designing that study. So while we agree that we may not need 7 PD-1, PD-L1s out in the marketplace, or even more that are currently being developed, yet at the same time, I think that we should think about the risk tolerance that companies are willing to take on when we actually are advancing innovation. And in that respect, when we do have 2 or 3 agents in the marketplace, there is life cycle management where we're looking at additional indications, looking at beyond, whether it's non-small cell lung cancer, colorectal cancer, pancreatic cancer, some of the rarer tumor types where we may not have pursuit of these. And if you have more than one agent, I think industry and different companies may pursue these additional indications.
Great. I think Gideon wanted to say something. Go ahead.
Yeah, no, I agree with what John just said, and everyone else, actually, Martha as well, and George. I mean, I think that if we're encouraging innovation, we really want, rather than a lot of copycat, Me Too programs. The reason to do a sort of non-inferiority study would be to see if you could either decrease the toxicity. If a drug is equally as efficacious but less toxic, I mean, that would be a good reason. And I know Laura is doing a lot of work on de-escalation of therapy. Can we get rid of anthracyclines, for example, in early breast cancer? So I think that's where the focus should be, rather than just flooding the market with Me Too's.
All right, I want to add to that and also go back to John's point. I think it's a great point about, do you want innovation or do you want to spend more resources in these, large comparative sort of efforts and studies? And I'll give you a great example of innovation with the PD-1. Okay, so when we were developing our PD-1, obviously Keytruda and also Opdivo were ensconced in they established in the lung and the melanoma fields where they had, major positions. And we were trying to figure out, well, what do we do? Can we do a head-to-head study? A non-inferiority study would be huge and so forth, all these things that we talked about. So what we did was we looked for a novel indication, a place where they hadn't been tried before. And we found, in fact, something that was astonishing to us, but that nobody had ever looked at, which was squamous cell carcinoma of the skin. And there, not only were we able to get an approval, okay, based on an innovative drug design that we co-designed with the FDA, but moreover, above and beyond that, okay, it was an area that had been completely missed by all the other players up until that point. And paradoxically, even though it was missed, it was the place where now for at least, solid tumors, it had the highest responsivity rate. And in fact, we've learned, I think, a lot about the biology and so forth and where to go forward with combinations based on those learnings. So I think John's point is a huge one, which is you got to watch what you try to solve for and what you design for. And there's always unintended consequences. But the current system does select for attempts to do that, which is innovation, to avoid, in some cases, having to go head to head in the existing indications and so forth. And there you get a big result. And there it's not just a new PD-1, it's a new whole indication and enormous benefit to patients who weren't receiving any benefit before. So I really want to echo John's points. I think it's a great point that you got to always watch what you try to design for, and the best intentions and so forth may end up leading to less incentives for doing the right things. And so that's what I'm saying. It's very complicated.
Great, thanks. So there's another question which kind of goes to the other end of the spectrum, perhaps. And that is, it's a question about using real-world evidence and using real-world evidence more often. Obviously, real-world evidence can potentially help with understanding of the impact of novel therapeutics in common diseases, particularly after approval. But the questioner kind of notes that there seems to be resistance with using that type of an approach, and was wondering what people's thoughts were on the use of real-world evidence towards, I think, kind of refining provision of therapeutic care. Any thoughts from anyone?
Well, I'll quickly repeat what I said before, and it echoed what Laura had said, which is nobody's against using real-world evidence. It's just that the quality of that data right now is not good enough for making a lot of important decisions. And we as a society have to decide whether we want to invest to create better real-world data so that then we can all use it and it can be used by society in some sort of large mega database that everybody would have access to. I think it would be, great to have that. But until we have real high quality data, we're a long, long way from really having useful real world evidence. And everybody has to understand that.
Yeah, I would just point out for those in the audience that the fact that you're hearing this from oncologists about use of real world evidence should make everyone take pause for a moment, because probably oncology is one of the places where real world evidence is probably got the most tailwind, right? Because you have endpoints here that are like life, death, alive, dead. That's a pretty good real world evidence endpoint. Duration of remissions, things like this. So it is an interesting place that we still have issues with real world evidence and challenges in this space. Gideon, you're nodding your head. Let me let you go ahead.
No, I totally agree with you, Peter. think, and what George said, we need to make the necessary investments because it is so wasteful that, there are these silos between clinical practice and research. And, we have whole departments who are trying to break down, that wall. So, I think we just, as a nation, as a society, we have to make the requisite investments, because right now, it's just no one's confident enough, from both from a pharma perspective and from a regulatory perspective to really make hard decisions just based on RWE alone in terms of approval.
And I would say it's not that it's that hard to do. It just takes the will to make a change in the way we do clinical practice. We are paid in clinical medicine to do things and record that we do things, but not, to systematically collect our outcomes as a part of practice, which I argue in 2021 should be an absolute essential part of our job. And if we standardize things, regardless of these EHR systems, and have something that's a modern technology that allows us to do this, it would transform the entire system. And it's high time that we make this kind of investment. And the FDA is actually investing in prototypes like OneSource and others. This is an absolutely essential thing we have to do to improve oncology care, or
John,
actually all clinical care.
Yeah, I agree with all the comments. And we've looked at some of the real world evidence, attempting to use it as additional supportive data for submissions and filings. And we really dug deep into some of the available data that was out there. It wasn't clean enough that we felt we could use for submissions and support. Peter, I'm sure you're familiar with that. So, but one where we actually have looked at, which is where we have alpelisib, which is a PI3 kinase inhibitor. And there is a PI3 kinase related overgrowth syndrome, which is a very rare disease with very limited number of patients. And you guys are probably familiar with that. We actually can collect that information. And I referred to this earlier, which is very rare populations. But we're working with specific investigators to go back and track down every single patient. If you're looking at a more common oncologic disease, I think that would be really difficult to try to track down all of these patients. But with PIK3CA related overgrowth, PROS syndrome, you can actually count on, maybe it's less than 100 patients where we can actually collect this information. So I think that these are the two disparities when we look at RWE.
Yeah.
Yeah, this is an interesting place, because what you just brought up, John, is really interesting, because it highlights something, an exact observation that I have, which is for really small populations and really large populations, real world evidence seems to be, because when we do vaccine effectiveness studies using like CMS databases in millions and millions of people, we can actually get results. And we actually have examples of, for instance, high dose versus standard dose influenza vaccine in that population where you can use real world evidence. And when you use really small populations in rare diseases where you can kind of pluck, you can get there. But for the kind of the general workhorse function where you'd like people to be working, we're still not there, which is, so it's not that it's I think it's almost like the promised land is like just over, it's always just over the border, and we just have to get there somehow. It's a great.
Yeah, and it's, we have to have the will to do it and provide the incentives to make it happen. This is a very doable thing. As George said, if we make it a priority, okay, then it will happen. Look at what we're losing by not changing the way we practice.
Yeah, I know, agreed. Great. Any other comments on that? There's a question here that maybe I'll direct to Martha to start with, and then others can add in. So there was a question basically adding, what can we do? Some of it might be learnings from the pandemic, but whether they're learnings from the pandemic or not, to help innovative companies move their products ahead more quickly. What do you think they need? What could we do more to help innovative therapies move ahead more quickly in terms of communication, perhaps from FDA or other things we could do?
That's a big question. I think there are lots of things that we could work on, certainly. I think thinking back to my experience with breakthrough therapy designation, I would have to say that where I think we tend to add the most value is through making ourselves more accessible and providing advice, not at just major milestone meetings, but throughout, throughout the course of development, trying to be as proactive as we can to kind of identify potential pitfalls along that process. And I think, the conversations that I've enjoyed the most and I think that have been most productive with sponsors have been the informal teleconferences where we have like a very specific question that we need to try to, bring our knowledge to bear on and kind of work through. And I think, having that mechanism for more informal interactions with industry to kind of move drug development forward, I think it's a small change. I think it really does seem to help streamline things and help erase misunderstandings and misconceptions that can sometimes really make drug development less efficient and effective. So I think having staffing and the ability to have sort of more open, more frequent communication, I think is really key. And I think also just working together proactively, the agency with stakeholders to help identify issues that we're running into, big issues that we can then discuss in a public forum, that aren't drug specific to try to tackle these problems. I think there are many things we can do. And those are just
anyone else have any thoughts from the company perspective? What could you, anything that you can think that you need from FDA?
Well, like I said before, I think that there are already, it's a great interaction and there's the necessary, balance and both sides have their roles and so forth. As I said, just from our COVID drug development, efforts, and it comes down to resource allocation. And I don't know if we can do this more broadly across all therapeutic areas, but right now the level of resource allocation and urgency and the timings and so forth, instead of 30-day turnarounds, there's 1-day turnarounds and so forth. I don't know if it's really possible and feasible to run a whole organization like the FDA across all therapeutic areas like that. I'm just saying I've been astonished at how the interactions and the turnarounds are, when you have this incredible degree of urgency and the appropriate resource allocation has been made there. And like I said, I don't know if that's something that's possible to be done on a more global level.
Yeah, Peter, I'll add, the resources that have been devoted to COVID has been unprecedented, and we've seen that turnaround. When we submitted phase 3 study design, sometimes we're getting them back within a week. And normally that takes much longer, as everyone knows. So it is really, truly unprecedented. And if we could get more resources at the FDA, we're big advocates for that, and that would be really helpful. And Martha, one other thing that you said that really resonated with me is the informal discussions. Often those are so helpful. It's so insightful when, because often we're designing these studies, we're second guessing and over designing studies because we don't know exactly what we're shooting for. So we try to target everything, and because we don't know what to address. And in that respect, we're trying to answer all the questions, and just a simple, quick response back gives us so much insight. So that's really helpful also.
Yeah, I would agree with John. Yes, I would agree with John on 2 points. One would be that, sort of the resources is very important, and creating that culture that exists in oncology at FDA. I think continuing to make sure that FDA clinicians and scientists get competitive salaries and continuing letting them publish and really push out novel research. I think all of those things help retain top people, because you need really smart clinicians and scientists for that collaboration. And then in terms of the seamless discussions and conversations, I think to embolden and enable FDA scientists, like when you truly have a breakthrough product, those discussions and insights are just critical, and not just to make sure that breakthrough therapies continues to to have for this all hands on deck mentality, rather than, you know, kind of every drug development program is you know, equal and should, you know, have equal amount of resources expended towards them.
Oh, that's great. I just to sum up, I think there's it's a lot of these answers really ring true here. I think this concept of early interaction, informal interaction, it does often make a huge difference. We see this in the cell and gene therapy field, where sometimes an informal conversation or two with a CMC expert at FDA, and that goes to Gideon's point, just to weave that in, you have to have really good people, because the people who are giving the advice have to give good advice. And that can make a huge difference and save time from going down the wrong rabbit holes, money from doing studies that are really not going to help advance the product, and ultimately get products to people in need more quickly. So points all well taken. And I think George's point is also really well taken, that the issue is, how do you allocate resources here? Because I don't think we'll ever get, we won't be able to continue. I mean, part of the reason, frankly, while we've been able to do what we're doing with these 1 week turnarounds, is because we have folks who are working 12, 15-hour days when they don't have to. And that's what's been, because they're of the incredible level of commitment at FDA to this, people are working ridiculous hours to do this. We won't be able to, you know, we can sustain this for a sprint, a COVID sprint, which is now turning into a COVID marathon. But I don't think we could have this become the new norm. I mean, I think that will have, unless we have additional resources. And I think that's something that we have to think about, because I think getting back to what John was noting, we've really realized that it's actually doing the public a disservice not to have those resources early on, especially for those informal interactions. So I think we have like 3 minutes left, and I wanted to turn it back to Laura, because I think she wanted to do 1 minute or
a lightning round
here.
All right, everybody. Now you've got 15-second answers. We have 2 quick questions for you. Okay, first, what new endpoints, and this is for everyone, what new endpoints and technology will be used to transform the pace of testing new agents in the next 5 years? Martha.
So I think use of artificial intelligence will probably really become, really help advances. I think circulating tumor DNA, hands down, I think is going to help. So,
Gideon.
the 2 big changes.
Echo Martha, ctDNA is here, metastatic and adjuvant setting, minimal residual disease for solid tumors. We just have to deploy it.
John?
Oh, those were my top 2. They grabbed them already. So I would say validated tests across sample types, blood, tissue, and technologies across protein, DNA, and RNA. Great.
George?
These guys said it all. You know, it's all about molecular markers and validating them going forward.
Peter?
I echo, and I also think I would echo what Martha said. I think AI on top of all of this may be a big boon.
Right, AI and ctDNA. Well, isn't that great? We all agree. Okay, last question. Okay, what lessons from the pandemic should we keep once the pandemic has passed? Okay, Gideon.
Rolling reviews, streamlined dossiers, mutual recognition inspections.
Okay, great. Martha?
Decentralizing clinical trials.
Great. Okay, John. Yeah, decentralizing. Okay, and out in the community, John.
Mindset, thinking about what assumptions that we hold and what's possible, working not just within industry, but collaboratively.
Yeah, great. George?
Well, I hope that it's not a lesson that we learn, but what society learns is that, collectively the entire ecosystem here has really, Paul touched upon it. At the FDA, they're working overtime. At every company, people working overtime, not for the sake of the almighty dollar, but to try to make a difference, for society and for humanity. And I hope this is a lesson that remains with the world, that, hey, we are such an important ecosystem, and we have to figure out how to resource it, how to invest in it, and incentivize it, so that we continue to get the sort of results that we've gotten with COVID? Is there any way to get those sorts of results even more broadly to take advantage of this incredible ecosystem that we have?
And more efficiently, no question. Peter.
So I think it's actually the continuation of global regulatory collaboration, the increased amount of harmonization that we've tried to have that we're working towards.
Yes, and I'll end by saying urgency. This is an oncology panel, so I would say that people with cancer have similar or higher rates of death than people critically ill with COVID. So we have to do things smarter and more efficiently and not lose that sense of urgency, and remember that people are dying every day. So we have to put our best foot forward. This has been a fantastic discussion, and we're ending on time, which is somewhat amazing. So thanks so much to everyone. And I'm now going to turn it over to Kuldev, who's going to introduce Peter for his keynote.